So let me first of all thank you for the invitation to come today and get a chance to catch up and hear about a lot of exciting presentations. I'm going to talk about animal models that we've been carrying out looking at the effects of, in particular, ambient ultrafine particles and their effects on brain development and behavior. So just starting out, we need to remind ourselves what air pollution is. This becomes important as we get further into trying to figure out the components of air pollution that are playing a role here. Obviously, we have primary pollutants, and the chemistry that goes on in the air generates secondary pollutants. These are primarily mobile and industry related, but I want you to also look at the right side of the slide for the ultrafine particle, where we're talking about a carbon core particle to which numerous contaminants adhere. And because these particles are so small, they actually have a greater surface area overall to which contaminants can adhere. And we talk about, obviously, air pollution in relation to particle size. US EPA regulates air pollution for what are called fine particles. Those are 2.5 micrometers and less, or coarse particles, PM10, 10 micron particles. Right now, data on ultrafine particles is still considered, has been discussed for regulation, but is still considered equivocal. It's also a much more difficult technical challenge. And I think on the left side, you get a sense of what the size of these particles are. The PM10 to 2.5 sitting on a human hair, and the PM2.5, sitting on top of PM10, and then the ultrafine 100 nanometer or less particles. So people have said to me, well, you know, the rates of neurodevelopmental disorders are increasing, so how could they be related to air pollution? Air is getting cleaner. Yes, we regulate levels of PM10 and PM2.5, and they've certainly decreased. And UFPs, however, despite the fact that they're considered the most reactive components of air pollution, are not regulated. And I think what's really been interesting in the last couple of years are studies that have looked at correlations of levels of PM10 or PM2.5 with ultrafine and actually don't find very good correlations, which would suggest that regulating PM10 and 2.5 is not necessarily regulating the levels of ultrafine particles, which are the most reactive component. So we know that the cardiopulmonary toxicity of air pollution is very well established. I'm sure most of you have seen the World Health Organization figures that list air pollution as the 6th leading cause of morbidity right now worldwide. The toxic consequences of air pollution on the brain are literally just being recognized. This research has really started only over about the last 5 years. And the figure of 6th leading cause does not include any of these effects yet on the brain. And most of these effects on the brain are being seen with respect to both developmental and neurodegenerative parts of the lifespan. So we need to think about how particles and or their contaminants can actually get to the brain during gestation. Ultrafine particle contaminants can cross the placenta and reach fetal brain. They can cause a direct maternal inflammation that affects the placenta and consequently the fetus. So multiple different mechanisms by which that can occur. After birth, there are also several mechanisms. Ultrafine particles in particular are so small that they basically bypass something like a macrophage, which would be there to pick them up. They get into the bloodstream, they are solubilized in the bloodstream, and the contaminants can then go wherever they want. One of the things that is a particular concern is that we also know that contaminants on ultrafine particles are actually taken up into the brain via the nasal olfactory route. So basically you are breathing these in. They are able then to bypass the blood-brain barrier. And so we don't necessarily know if we take a peripheral blood sample what's going on in terms of the brain, because these have bypassed that whole system. I think the other thing to be thinking about is that the vagal nerve, which finds its way directly into the brainstem, is probably another route of uptake of particles directly into the brain. Okay, so epidemiological studies, we now have something like 15 plus studies that have found an association of air pollution metrics with autism spectrum disorder. There are studies reporting effects related to schizophrenia outcomes, a growing literature on attention deficit disorder, a slower rate of cognitive growth, and what's called interhemispheric disconnectivity, which means the two sides of your brain are not connected the way they should be by the white matter of the brain. There are, as I'm sure most of you know, increasing rates of autism spectrum disorder, and the rate at which this rate is increasing cannot be explained solely by genetics. And so people have begun to start looking at environmental contaminants. So what does autism spectrum disorder look like? It is diagnosed on the basis of behavioral features, social communication deficits, repetitive behavior, inability to inhibit responding. It can also include sensory motor deficits, cognitive inhibition, and there are several neuropathological features that you'll see, and I think I show you a few examples of them. They're actually dynamic over the lifespan. The brain starts out being too large, but as time goes by, it's actually smaller than a typically developing child. The brain starts out with too much myelin, too much white matter, but over the course of time, it actually falls below that of a typically developing child. Something called ventriculomegaly. This is enlarged lateral ventricles that I'll show you. There's inflammation lifelong, mitochondrial dysfunction, and what's called excitatory inhibitory imbalance, which is basically too much glutamate. And these basically correspond to some of the hypothesized mechanisms for autism that you see on the right-hand side of the slide. So one of our questions has been, how much plausibility do we see from experimental animal models of developmental exposure to air pollution? I'm going to talk about work that we've done looking at postnatal exposures of mice to concentrated. They're concentrated about 10 or 20-fold ambient. We pull these in from outside, separate them by particle size, and deliver just the ultrafine particles. So they still include all of the contaminants. They don't concentrate the gases. And then I'm going to talk about gestational exposures. These are all in mice to concentrated ambient fine ultrafine particles that was carried out at NYU in Tuxedo, and now unfortunately that facility for ambient exposure is no longer available. So this is just a schematic showing you our intake of air from outside on a very heavily trafficked road at the university. We pull it in, separate it by particle size, and then deliver it in another room, exposure room. We call it concentrated ambient ultrafine particles, CAPS. So you'll see CAPS in all of these slides versus filtered air controls. This is the scheme that we use for exposing during the postnatal period. We expose animals, I'm sorry, this is a little bit, the slide is a little bit moved, the days. We actually expose animals from days 4 to 7 and 10 to 13. We do these exposures for 4 hours a day at the beginning of the day when the traffic is the highest. Some of these animals undergo behavioral testing. We take tissue at various times throughout the course of these. What's important to remember is that the rodent, the mouse, is different from the human because its third trimester is really this early postnatal period. So the period that we're looking at here is the human third trimester brain equivalent. It is a period that is marked by rapid increases in neurons and non-neuronal cells within the brain. So it's a very active period of brain development. And from a exposure point of view, because we use ambient air, we're actually at the mercy of whatever is going on outside that day. And so I'm going to show you studies that occur at different mean particle counts or mass. The first ones that I'm going to talk about were at a level of about 100 micrograms per cubic meter. And the slide on the right just shows you that our particle diameter across these exposures is always less than 100 nanometers. So these are ultrafine particles. What does that look like relative to other places in the world? That first exposure, about 96 micrograms per cubic meter, is high but not irrelevant, as you can see, in contrast to probably not so bad day in Beijing and measures that have been made in Los Angeles. So what I'm going to show you is that the changes that we see in mice are very consistent with hypothesized mechanisms of autism. We see ventriculomegaly, we see aberrant development of myelin, white matter tracts, the two sides of the brain being essentially disconnected. We see lifelong inflammation, microglial activation, elevated levels of excitatory inhibitory imbalance, too much glutamate. We see perseverative and repetitive behavior. And these are either seen only in males or they're seen to a greater effect in males. This is what a mouse brain normally looks like. You can see at postnatal day 4, that area of white in the middle is the lateral ventricles right here. By postnatal day 14, those ventricles should be closed because all of the brain infrastructure that develops over that period of time basically begins to squeeze down the sides of the lateral ventricle. And this was really my introduction to the neuropathology of ambient ultrafine exposures. In the top, you can see brain sections from a filtered air control. And I think in the bottom, you can see quite clearly that those lateral ventricles are still very much expanded. This is something called ventriculomegaly. You would see it in a child, sometimes in things like periventricular leukomalacia. Over the top of the ventricle is an area called CC, corpus callosum. That is the largest white matter tract of the brain. It extends from the front all the way to the back, and it basically connects your two hemispheres. You can see that it is largely, it's got a lot of holes in it. It's very tattered, if you will. So not only ventriculomegaly, but clearly disruptions in white matter development. This is something that we saw all the way out. We don't see this in females, we only see it in males. And I think if you look at the right-hand side of the slide, you can see that it's still there at postnatal day 270 in these mice. That was the last time point that we examined to date. This is the size of the corpus callosum, that white matter tract. You can see in males in panel A at 14 days of age, so right after exposure ends, the corpus callosum is reduced in size. It does catch up in size by postnatal day 270, at least in terms of mean values. But it's myelination, the development of the myelin in that corpus callosum is severely reduced. You can see on the left side in the orange how much of that corpus callosum is stained by something called myelin basic protein, which gives us an indication of the myelin levels, whereas on the right side you can see a mouse that was exposed to CAPS, to the ultrafine particles, where the extent of myelin is at about 15% of the total area of the corpus callosum. This aberrant corpus callosum white matter development tracks the white matter development along this structure. So you can see in this at postnatal day 14, huge reductions in myelin in two areas of the corpus callosum. One's called the truncus, and the medial external capsule. These are the parts of that that are actually developing during this period of exposure, where the lateral external capsule is spared and develops after our exposures are ended. We also see microglial inflammation, activation that appears to be lifelong. Again, this was something that was male-specific. Here you can see it on the left side in an area called the anterior commissure at 14 days of age. and in the hippocampus at 55 days of age. And then if we take a brain from postnatal day 270, so the latest time period we looked, you can see that in the corpus callosum, there's still a very large increase in microglial activation. It's literally 340% of control. And these exposures, remember, ended at postnatal day 14. We see an excitatory inhibitory imbalance, basically too much glutamate in these brains. There is a slight trend for that to happen in the females, but it is statistically significant only in the males. And that, again, persists at least as long as we have looked at it. Behaviorally, we were interested in measures of cognition, measures of behavior associated with autism-like phenotypes. One of those is impulsivity, inability to inhibit responding, making impulsive choices. This is a paradigm that we use in humans called self-control or delay of gratification, where we give you a choice between a short delay, but you only get a small reward, or a long delay that's followed by a much larger reward. We made a slight variant of that in which we told mice, okay, you can get free rewards. They're just going to come, free food deliveries over and over again. First, you have to make 25 responses. This is in an operant chamber Skinner box. And once you make the 25 responses, you start getting free rewards. The trick is here that the time between free reward deliveries just keeps getting longer and longer and longer. And so then you can make a response, reset the 25 response requirement, but go back down to the shorter delay values. And this is just a schematic of what we call a cumulative record of what behavior looks like for a representative air animal. Makes its 25 responses, then starts picking up free reinforcers. You can see that there are long periods of what we call waiting behavior there, where the animal is getting free rewards. If you look at the bottom, this is an ultrafine particle exposed animal. You can see that waiting behavior is almost non-existent. Those would be the horizontal parts of this, and they're resetting this over and over again. So at the end of the day, their mean waiting time is very, very short, and the number of resets that they make is very large. So in essence, they're making many, many, many more responses for each food delivery that they ultimately get, which is very contrary to what we like to see from an evolutionary perspective. We carried out another exposure where we averaged 46 micrograms per cubic meter, so far lower, as you can see here in that study. We still see ventriculomegaly in the males. As you can see here, these shouldn't be treatment, they should be sections along the corpus callosum, but it's still there. We're still in the process of looking at some of those tissue. We've also looked at those animals in some very specific social behaviors, preference for a novel conspecific versus a familiar conspecific. So preference to engage with a novel mouse versus the one that you live with, and you can see a picture on the right of how that works. And that is reduced. The preference for a novel male is significantly reduced by these exposures, but not the preference for your cage mate. That was related to the levels of testosterone, which are in fact reduced by developmental air pollution exposure. So lots of possibilities about what could be going on here. Another talk by itself. There are a lot of different mechanisms of development going on during this period of time. We haven't really had a chance to fully look at the role of sex hormones in this, as well as what mechanisms of inflammation, but we'll come back to this. So what about gestational CAPS exposures? This, for the mouse, is essentially trimesters 1 and 2. And these were data that were part of a collaborative study, again, where the animals were exposed to ambient fine and ultrafine particles in Tuxedo, New York, from gestational day 0.5 to 16.5, and we collected brains from these animals. These exposures were very similar to the first exposure that I talked about in terms of their mass. They were about 93 microgram per cubic meter across time. They do not produce any kind of overt toxicity or reproductive effects at all. What you see under those conditions, again, is ventriculomegaly. In this case, though, we see it in both sexes. Remember, this is a very different period in terms of brain development and the kinds of where we are in the trajectory of events. You can see in the bottom that we see this across the ventricles, all of the sections that we looked at going from lateral to medial parts of the brain. It is persistent, in this case, in the females, but not necessarily in the males. So the top panel shows you that data again from postnatal days 11 through 15, and the bottom is postnatal days 57 to 61, and those were the latest time points that we had for those brains. So that ventriculomegaly persisted in the females in this case. We also saw in both sexes small but nevertheless significant reductions in the size of the hippocampus. And unlike what you see with postnatal exposure, where the corpus callosum size is actually shrunk down, in this case, it's way too big. It's much larger than it should be at this point in development, as you can see in the bottom two panels, and this was for both females and males. The other thing, not only was the corpus callosum much larger than it should have been, it had way too much myelin, and that was happening way too early. You can see at postnatal days 11 through 15, that increase in the, this is actually in the area of that, and the figures look exactly the same at postnatal days 57 through 61, both for the size and the myelination in the corpus callosum. There is an inflammation that goes on, and here we're looking at the number of microglia. These are can be inflammatory. They are actually required for neurogenesis and gliogenesis to go on, and they have a different activation state. So in the bottom series of 4 panels, you can see a quiet, what we call a ramified microglia, all the way to a very activated amoeboid, number 4. And I think if you look at the profiles for both males and females, there are increases in the activated, the number 4 amoeboid size relative to filtered air control, and even a slight decrease in the number of microglia in these brains. The other thing we've started doing is looking in more depth. This is a myelin sheath damage. So they had too much myelin, as I explained before, which turns out not to be due to the caliber of the axon. So it's not like the axon is too big. It's just that there's a lot more myelin wrapping around it. But at the same time, where you see these yellow arrows, what you see are actually holes in the myelin. Not like the myelin is unwrapping, but literally holes into the myelin itself in both female and male brains that are exposed to ultrafine particles. So one of the things that we've gotten into is asking the question, because it comes up in every grant review, what are the components of air pollution that underlie its developmental neurotoxicity? There are so many contaminants on these particles. There's inorganic elements and metals, lots of organic contaminants, polycyclic aromatic hydrocarbons, endotoxins, or what are called LPS, widely used as an inflammatory model for neurodevelopmental disorders, and there are gases as well, and the chemical interactions of all of the above. So we've already looked at, we've generated pure ultrafine carbon particles. So we basically generate pure ultrafine carbon. It doesn't have any contaminants adhering to it. And we've also generated LPS. These are endotoxins, atmospheric endotoxins, exposed animals to either or both. And we don't actually see much of a replication of the phenotype that we see with the ultrafine particle itself. So we turn back to what causes overmyelination. And of course, myelin is really the result of iron availability. Iron is what drives myelination in the brain. That process isn't really all that well established in rodents until later in postnatal development. So there's a very long period of time during which iron can actually reach the fetal or neonatal brain. So we've analyzed filters from these exposure chambers for trace elements, and you can see those here. And something that I hadn't even thought of before, the major component there is sulfur. And you can also see that, especially for the gestational exposures, the second largest relative quantitation-wise is iron. We see iron in the postnatal exposures from Rochester as well. And so one of the things we started to do is to look at the iron and sulfur content in brain. These are animals that were gestationally exposed. We look in the olfactory bulb because we're very interested in where these are going. And you can see in the females, there's an increase in iron. This is the bottom left, increase in iron in the, I believe this was in the corpus callosum. And then if you look on the right side of the slide, there's a threefold increase in iron in the cerebellum of both sexes in this case. So way too much iron in the brain, way too early. You can also use high-resolution transmission electron microscopy. These were also from gestationally exposed animals, and when you begin to do that, you can see elements of iron, silica, and aluminum in these brains, all of which you're going to see on the filters. You can see on the far right that these metals tend to agglomerate into larger size particles. This is a 50 or 20 nanometer picture. We've also used ICP-MS, a method to look at metals across these brain sections. This is from postnatal exposures, and I'm showing you the levels of iron and sulfur in these brains from postnatal day 14. You can see that there is just marked increases in the levels of iron and sulfur. They are seen across the brain, which is consistent with what you'd expect because of the functions of both iron and sulfur in the brain are widespread. They're related to antioxidant production and mitochondrial function, which is all over the brain. This raises sort of a new interest in something called ferroaptotic mechanisms of ultrafine particle damage, where there's too much iron going in to a cell that ultimately would be taken care of by glutathione peroxidase, but that is also reduced under these kinds of conditions, so you don't have the mechanisms in place to do this. This is now well established in neurodegenerative disease, but nobody has really yet looked at this with respect to neurodevelopmental disorders and not to air pollution either. Sulfur is critical in the brain. You don't necessarily, I never really thought about it, but it's involved in the production not only of glutathione, which is the major antioxidant, but also for glutamate production, and these things interact. And iron and sulfur form cluster proteins, which are also important to this function. So sulfur is very important. I hadn't thought about it. Actually, reductions in glutathione are considered a biomarker of autism spectrum disorder, and reductions in things like glutathione, which are produced by air pollution, by iron exposure, and by sulfur, are also seen in other neurodevelopmental disorders. So I just want to close by reminding us, I talked a lot about this relative to autism spectrum disorder, but while each neurodevelopmental disorder tends to have unique features, they also share any number of features, which I list here, whether it's reductions in white matter, whether it's inflammation, ventriculomegaly. We'll see these in lots of different neurodevelopmental disorders. And so if this developmental air pollution exposure is a risk factor, it's going to be broader than for something like autism spectrum disorder. I think the other thing that's interesting is that air pollution exposure is incredibly heterogeneous for any individual on any day, any hour of the day. And if we now superimpose this over the course of pregnancy, that means that pregnant individuals will have different exposures at different times. And all of these neurodevelopmental disorders, and particularly autism, are highly heterogeneous in their phenotypic expression. So if we sort of think about the heterogeneity of the exposure, in a way it very well matches with the heterogeneity of the phenotype. So just to close, exposures to real-time, real-world, human-relevant levels of ultrafine particles during periods of brain development can produce features that are reminiscent of autism spectrum disorder and other neurodevelopmental disorders. They differ by sex. That's consistent with the fact that brain development is different by sex from cell 1, and they differ by developmental period of exposure, what we would expect. These effects are seen in multiple human neurodevelopmental disorders, again, suggesting that we have to think more broadly than just something like autism. If we start looking at we need to start looking at the components of air pollution that are responsible for these effects for multiple reasons. One, we'll be able to build better animal models. It'll provide us with a much more focused strategy for looking at mechanisms and for intervention strategies. And with respect to public health protection, it may be that we need to regulate levels of iron and or sulfur in the air. So with that, I just want to thank all the individuals that have been part of these studies, in particular my colleague Günter Oberdorster, who is Mr. Inhalation Toxicology, brought you ultrafine particles, who have made these studies possible. So thank you very much.